normal divisions of the cells, about the meiosis. Guys, keep quiet. Keep quiet. Coming. I told you about the normal divisions, about the prophase, metaphase, anaphase, and telophase. And you are seeing at this slide, we have abnormalities in chromosomes. We can have this abnormality in numerical and structural. Numerical means at the number. The number of the chromosomes being changed. And the structural, it means that the structures of the chromosome being injured or being damaged or being disrupted. In the numerical abnormalities, we have three types. You are seeing Turner syndrome, trisomy 21 or Down syndrome, the name is similar, and another syndrome, we call it Klinefelter. So Turner, Klinefelter, and trisomy 21. These are the numerical abnormalities. It means that in some situations, instead of having 46, we have 47, we have 45, okay? These are the abnormalities, and also the structural abnormalities, we will see. The first one, Turner syndrome. I told you we have 46 chromosomes. But at this syndrome, at this disease, we don't have 46. We have 45. We have 45. Okay? And also, it is happening in females, not males. Okay? So it's just related to the females. And instead of having 46, we have 45. So what happened to one chromosome?
Disappear?
Yes, it's disappeared based on the divisions. For example, I told you in the meiosis, we must have, for example, we have the divisions and we have 4 cells. Okay, the polar bodies and the primary oocyte. Okay, this one, which wants to take part in fertilization, it doesn't have any chromosomes, disappearing or lost or missing. What happened, and you are seeing this view of this syndrome. We call it Turner syndrome. So it is 45 instead of having 46. In the next slide, you are seeing trisomy 21, Down syndrome. Look at here, look at this map. All of them must have how many? 2. Okay? But look at this one, the chromosome number 21. We have 3 chromosomes. So we call it Down syndrome or trisomy 21. It means that in the chromosome number 21, we have 3 chromosomes instead of having 2 chromosomes. And usually, so we have extra chromosome. So it is 47 instead of having 46. Okay? And also I told you in the 21 chromosome, and if the person, because in the Down syndrome we have it in the women's and also in the men's. In the women's it is 47XX, and in the men it is 47XY. Okay? And I told you instead of having 2 copies of the chromosome 21, we have 3 copies. And you are seeing the picture and the morphology of this trisomy 21 syndrome. The next one, Klinefelter syndrome. It shows himself in men or women? Male. What happened that this Klinefelter syndrome is happening? We have 1X extra. What does it mean? I told you.
Basically, if a boy comes with excess Y chromosomes, basically they would have like gynecomastia, wider ovaries, produce secondary sexual characteristics like pro-obiotic, all of that. But like usually it's not like not all of his somatic cells are fully XXY. Like the amount of like the severity of the symptoms depend on how much of the somatic cells are affected by this. And also there's another one, but like for females.
We have just Klinefelter is occurring in the men's. If it happening similar to this one, happening to the female, it has another specific names. Okay, so I bring for you, we have lots of numerical abnormalities. I just bring three usual example, okay, for this disease. As your friend said, we have an extra X. I told you, we have 46 X, Y in men. But at this syndrome, we have an extra X. So, and instead of having 47, we have, instead of having 46, we have 47. You are seeing 47 X, X, Y. An extra X. And what's happening? You are seeing the morphology of this syndrome. Your friend said true. We have gynecomastia. It means that the breast of a man is bigger than the normal. Another thing, it's lower IQ. Okay? Lower IQ. It's tall. The leg is tall. The muscular tone, the ability of the muscles is good. Okay? So these are the things that we have in men with Klinefelter. And also reduce the secondary sexual characteristics. In the men's, it's normal that the men's has loud. They are speaking loud. Okay? But the female are speaking a little low. But this person doesn't have these characteristics.
The concentration of testosterone is low.
Because of?
Testosterone.
Because, yes, that's true. Because it's related to the hormones. It doesn't have, for example, they don't have hair in the bodies. Little hair in the bodies. We call it secondary sexual characteristics. Okay? So you are seeing, and lots of them has infertility. It means that they cannot make the babies. Okay? They have infertility. These are the three numerical abnormalities in the chromosomes. After that, we have structural abnormalities. I told you, we can have lots of chromosomes. Instead of having 46, we have 45 or 47. But we have structural abnormalities. Angelman syndrome, Prader-Willi syndrome, and Miller-Dicker syndrome. We will see them. The first one, Angelman syndrome. You are seeing this girl. What happened that this Angelman syndrome happening? Look at here, inactivation of one gene. The name of the gene is not important. What one gene, I told you, the gene carrying the information. And when the one part of the DNA, we call it gene, is inactivate, causes this part. Which part of which chromosome? One part from the chromosome number 15. Okay? So inactivation of the gene in chromosome number 15 leading to Angelman syndrome, but not in the men. In the women or maternal part. If we have inactivation in the chromosome number 15, but in the male, this thing happening. Prader-Willi syndrome. Both of them, Prader-Willi and Angelman, has abnormalities in chromosome number 15. One of them in the man, another one in the women. Okay? So what's happening in this syndrome? You are seeing delayed motor skills. It means that the person, the child with this syndrome, we know that, for example, at age 3 or at age 2, they are starting to walking, and for example, at the age 7 or 6, they are starting to painting and writing something. A skilled movement. Okay? They have delay. And also they have inability or delay to speak. Okay? They cannot speak very well. They are always happy. It's not bad. Okay? But they are always happy. They are all smiling. And also white mouse. White mouse and being happy. And it has microcephaly. Cephal means related to the brain, related to the skull. When we say microcephaly, it means that the brain is small. And also we have macrocephaly. It means that the brain is big. It's not good. It doesn't mean that when you have a big head, you are very clever, you are very wisdom behavior. No. It's just related to the synapses between the neurons. So the Angelman syndrome, I continue, and they have a sleep disturbance. They cannot speak very well. So if one person cannot think, cannot speak, and being happy, it's not acceptable for us. So this is the inactivation of chromosome number 15 in the female or maternal part.
So all of it is mental issues.
Yes?
Mental issues.
Yes? Yes. No. Both of them. Mental and also functional, because we are seeing that they have delayed motor skills. Maybe they examine, it shows that maybe they have problems in the muscular part, and also they have inability to speech. If we have one disability, maybe it's related to the muscles and nerves, and maybe related to the brain. Okay, it has several factors. Okay, they must be discussed about them. So these are the main features of this Angelman syndrome. The next one I told you, Prader-Willi syndrome. It's again in the chromosome number 15, but it's not in the maternal. It's related to paternal or to the father, to the man. Okay, and you are seeing, and it affects to the growth, metabolism, and behavior. You are seeing the view of Prader-Willi syndrome. It means that if you have disability in chromosome 15 in the female, you are happy, you are good looking, but it's happening in the man, you are seeing with this view. Okay? So the next one, Miller-Dicker syndrome. Look at here, 17p13. What does it mean? It means that it occurs in chromosome number 17. And also, each chromosome for example, this is the chromosome, okay? Each chromosome is something like this, okay? They are attaching at this part, and also I told you we have two chromatid sisters. One part is big, another part is small, okay? We call it small brachium and large brachium. This one is P, and this one is Q. If you see, for example, in some books, in some disease, when you are studying, one thing happening to P, it means that short arm, this one. This is small and this one is large. So it shows that the Miller-Dicker syndrome, and also this is, for example, this is the 17, chromosome number 17. It means that it's happening at this part. And also not all of this part. We called it, for example, 13. It means that it's related to the region and bond. They are very specific. It's not, it doesn't, you need that for knowing about that. But 17 means related to chromosome number 17. P means related to the short arm. And this one, the 13, is related to one part of this short arm. So what happened in Miller-Dicker syndrome? This is affecting in the brain development. So it is again mentally, okay? And you are seeing that they have microcephaly, and also I told you about the deletion on the chromosome of this number I told you. This is the Miller-Dicker syndrome. After the Miller-Dicker, after the numerical abnormalities and also structural abnormalities, you are seeing the gene mutations. What does it mean, mutations?
organizations of the genes, change in the organization of the genes,
Succession of the gene.
Yes, both of them true.
There is a lot of types of mutation, for example, changing the number of chromosomes,
Mhm.
genes, the locations, and this stuff. All
That's
these
true.
mutations.
Changing in the gene.
Nucleic weights. Nucleic weights. Like,
Yes.
copies of,
Because the gene is a part of the DNA and it's contained the nucleotides. Okay? So changing in the gene, we call it mutations. The mutations in the genes can be dominant, and can be recessive. What does it mean? Look at here. For example, we have these two genes. Okay? We have one of them from the mother, one of them from the father. Okay? What happened in the child? If one mutation in the gene is dominant, it means that when one of them has the mutation, leads to disease. Just one of them, making disease. But in the recessive, look at here, we have one in one part, but it doesn't lead to disease. When we have the recessive mutation in the genes, we need both of them, mutation in both of them. If we have just one, it doesn't lead. It's just not disease. We have these genes, but it's not showing himself.
Like sickle cells anemia. Sickle cells.
Sickle
Sickle cells.
So it's not showing himself. You just have this information. You just have these mutations, but it doesn't show himself. But when we have dominant, if we have one of them, leads to disease. Okay? So this is the differences between dominant and recessive mutations, and you are seeing that for genetic factors, we have two copies, one of them coming from mother and one of them coming from father. For example, it's coming from mother and this one is coming from father. The mother is completely healthy. The father has the mutations. So it leads to disease because it is dominant. It's showing himself. Dominant means very powerful. But in the recessive, if one of them has the mutation, no disease, and it must have both of them, mother and father, have this mutation, which leads to this one, which leads to disease. After this primary information, we will go to the main things in embryology. Oogenesis. What does it mean?
formation of the cell, of the egg cell, of the ovum.
In men or female?
In the female.
In the females. Okay? So it is a process. When the first cell or precursor cells, we call it oogonia, which comes and differentiate in one process, one important and complicated process, into mature oocyte. After that, this mature oocyte participating in fertilization. Okay? So we called it oogenesis. You are seeing it's starting from PGC cells, I will tell you, primordial germ cells. These are the precursor of all the sex cells. Okay? And they are coming to doing the mitosis, several mitosis, and after that it goes to the meiosis, and after that meiosis 2, and at the end we have this part, ovum and the polar bodies. The polar bodies are not functional. But this one, the ovum, is functional for us and participating in fertilization. The PGC. I don't want to say about the PGC at this session and also the next session. But after that, because I must tell to you, we have epiblast, hypoblast, the cells from the epiblast and hypoblast coming and making these primordial germ cells. But at this time, just memorize the name. Primordial germ cells. at the top of this circle. Primordial germ cell. I will tell you what is this primordial germ cell. This primordial germ cell, imagine that you know about the primordial germ cell, okay? It's made, and they are coming to the gonads. Gonads of the embryo which is making. And it wants to differentiate in ovogonia. By means of what? By means of several divisions. Mitotic and also meiosis divisions. Look at here. At the end of the third month, it means that the embryo is making, and 1 month passed, 2 months passed. At the end of the third month, here, this is the surface, this is the ovary of the female. Okay? Look at this part. And the divisions is made, and we have primary ovocyte based on the mitotic divisions. From the ovogonia, we have mitotic divisions, and we have primary ovocyte. This primary ovocyte, you are seeing that these are covering by the cells. I told you these events are happening in the ovary of the female. Okay, and we have in the ovary, we have, for example, we have surface epithelium, we have connective tissue. This surface epithelium making these cells. Look at here, I remove this one. You are seeing these ones. They are covering all of these cells. Okay, we call it epithelial cells. So these epithelial cells are covering all of the cells here. And these are making by means of surface epithelium. So you have, for example, you have a class and it has several epitheliums, just like these valves. They are surrounding the class. And here in the ovary, we have the epithelial cells, epithelium of the ovary, which is starting to make this one, flat epithelial cells. These flat epithelial cells are very important. This one, flat epithelial cells. Why these are important? Because they are covering or surrounding the ovogonia. You are seeing. This ovogonia is starting to grow and divide to make what? Get here? This primordial germ cell making this ovogonia, and this ovogonia is starting to divide, and at the end we have ovum. So the primordial germ cell is making, going to the gonads, and after that starting to mitotic divisions. At the mitotic divisions, we are making the ovogonia, and this ovogonia is covering or surrounding by means of the flat epithelial cells. So the ovogonia is made, and they are surrounding by means of the epithelial cells. And this ovogonia is starting and also starting the mitosis. And what happened? We have primary ovocyte, this one. So at first we have these ones. They are starting to grow and divide, and after that we have the primary ovocyte. This primary ovocyte is starting to meiosis 1. So before the primary ovocyte, we just have mitotic divisions without reducing the number of the chromosomes. Okay, understand? So these are making, they are acting by mitosis, and they are making the primary ovocyte. Before the child coming to the world, they are arresting or they are stopping at here. Prophase of the meiosis 1. They are arresting or they are stopping at the prophase of the meiosis 1. You are seeing primary ovocyte arrest or stop in the prophase of meiosis number 1. What happened after that? You are seeing maturation of oocyte before the birth. I told you, they are starting to mitosis and meiosis. But the meiosis doesn't develop. I told you we have meiosis 1 and meiosis 2. But the primary oocyte step at this part. Doesn't want to go to the next step. When the next step is happening,
Puberty, when
yes.
luteinizing hormone is released, it makes like pre-ovulation go.
Yes.
Which makes it like finish meiosis 1, and then meiosis 2 like isn't completed until fertilization is
Yes.
done.
So we have stopped at the prophase of the meiosis 1. And we have prophase, then it's coming to the word again, prophase of the meiosis 1 until puberty. When the puberty is happening, it's completing the meiosis 1. And again, I will tell you, it's stopped at meiosis 2. And when the fertilization is happening, it's going to complete the meiosis 2 and the rest of it. So, look at here. These primary oocytes, I told you, they are surrounded by the cells, which coming from the epithelial cells. Okay? So, we called this one, this view, we called it primordial follicle. What does it mean? It means that the primordial follicle contains primary oocyte plus the surrounding layer. So you have the oocyte in the prophase of meiosis 1 and one surrounding layer coming and this one. Look at here. We have the primary oocyte and it's covering by means of the epithelial cells. So we call this one as primordial follicle. The primordial follicle contains primary oocyte plus surrounding flat epithelial cells. Understand? Yes or no? Yes? Completely yes? Okay. So, again, look at the picture. We have the ovogonia, which comes from the primordial germ cells. They are starting to mitotic divisions. Then we have the primary oocyte. At the first time, they just covering in the previous slide. They just covering by means of one layer. But when it's growing, this surrounding layer being specific for each primary oocyte. So we call this view as primordial follicle. If I ask you at the exam the primordial follicle, in the primordial follicle, the oocyte is in which stage? You must say that it is stopped at prophase of the meiosis 1. Okay? You go and take a break, then we continue at the 10. We continue.